Welcome to this webinar on early development challenges and solutions. My name is Nick Myers and I lead the product development and program management team at Boyd Consultants. In today's webinar, I'm going to highlight some of the most common challenges that face groups and organizations developing drugs and during the very early stages, and how they can be overcome or avoided, be they small molecules, traditional biologics, or advanced therapies. And as we go forward, I'll try to offer some tips and ideas about how to avoid some of these issues. First, a very few words about Boyd's. We were established a little over 15 years ago, and we provide strategic advice to clients operating at different stages of the development pipeline. We work with clients developing all types of different products and drug and medical device combination products. But during the past 7 to 8 years, we've begun to specialize more and become known for our work on cell and gene therapy products. And we've worked on 9 of the so far approved cell and gene therapy products in the US and Europe. Boyd's is growing, and we currently employ 40 consultants in the UK, Ireland, and in the United States. Arguably, where we achieve our greatest impact is in helping startups and spin-outs at the early stages of their development journey, who are developing products largely via outsourcing. I think it's important at this point to say, why do we do what we do? Well, ultimately, we want to help to bring new cutting-edge treatments to patients, and we try to impact the high attrition rate of medicines in development. And we help our clients develop their products as efficiently as possible, help them to de-risk the product and avoid pitfalls later on, and to generate the data needed to support their partnering efforts for later stage development and commercialization. Apart from the development of the vaccines for COVID-19, which arguably was a special case, the development of medicines is generally a lengthy process. On average, it takes 10.5 years from approval of the first phase 1 study to launch of the drug. But this doesn't also take into consideration the transition of the drug from the candidate selection stage at the end of the discovery phase through to completion of the IND or CTA enabling package, which can account for a further 3 to 4 years. So the process is lengthy and highly complex, and different specialisms and different expertise is required along the journey, some throughout the whole of the development life cycle and others only during parts of the process. Let's now take a look at the product development pathway in a bit more detail, and I'll highlight some of the key points that need to be addressed at each stage. So firstly, candidate selection. When we're going to select the candidate to take into development proper, we need to understand enough about the physicochemical properties of the molecule and its ability to be formulated and delivered by the intended route of administration. We need to know about the extent of target engagement, which tells us about the potency of the drug, and its degree of selectivity, which may give clues about whether we're likely to encounter off-target effects and any overt safety issues. We need a basic understanding of the PK and the ADME profile of the drug, and I'll talk later about the non-GLP in vivo studies that one can usefully conduct at this early stage to help make a go or no-go decision. Next, we proceed to the preclinical development proper, which is where we initiate a GLP, IND, or CTA enabling package, which is expensive and time-consuming. Therefore, it's important to ensure that this is designed correctly and we have the right package of studies to support the route of administration, the dose level or levels, the intended duration of dosing in the clinical indication or indications. And for this, one needs an outline study design or designs for a first-in-human study and or initial patient studies. And one ideally needs to sanity check the intended pivotal preclinical program with the regulatory agencies at scientific advice or pre-IND meetings. At the initial clinical stage, first-in-human studies with small molecule drugs have traditionally been in healthy subjects and combine single ascending dose, multiple ascending dose, or multiple dose designs with the ability to measure PK, the effect of food, imaging, and sometimes drug-drug interactions, all within an umbrella protocol. With the advent of more and more advanced therapies being studied in rare diseases, the first in human study now is very often in the patient population itself. And it's very common to see phase 1A, 2A combined studies with a dose escalation phase, first of all in the patients, and then a cohort expansion phase in a larger number of patients. The next phase involves larger clinical studies, phase 2, 2B, or phase 3, which is about confirming the efficacy of the product. So unless the indication is a rare disease or there's a huge unmet medical need with no approved treatments, this usually requires 2 appropriately controlled phase 3 clinical studies to support a marketing application. The final stages of the pathway involve the preparation and submission of the marketing application and the review and approval process, which can be very long and complicated processes and need careful and detailed planning and management to deliver successfully. And many products still fail or are significantly delayed in reaching the market at this stage. The overall failure rate during drug development is around 95%. So only 5 or 6% of drugs that start the development process are being approved and reaching the market. The reasons for this are multifactorial. Toxicological findings in non-clinical studies account for around 20% or so of the failures. Lack of efficacy for another quarter of failures, or other clinical safety concerns, which account for 12% of failures during later stage clinical development. There are other reasons as well why drugs can fail. These can be to do with inability to manufacture at the commercial scale or other technical issues, cost of goods, which can be too high, commercial viability or issues around the external environments and competitor activity, or a lack of funding to complete the development program, or problems with the intellectual property portfolio. Drug development is expensive, and done properly, one cannot cut corners. In fact, it's critical that we don't cut corners because it reduces the likelihood of being able to partner and increases the probability of having to do rework. Early stage companies need to raise money at various points to fund the next phases of development. And their ability to do this is influenced directly by the quality of the development planning, providing realistic timelines and costs and robust development plans designed to meet the required target product profile, and engagement with major regulatory agencies is perceived to de-risk the program. Also, having robust intellectual property protection and being appraised of the competitor landscape is critical so that we can understand how the new therapy will fit in with current treatment paradigms with standard of care now, but also the projected standard of care and treatments which will be available at the time of launch. It's critical to build a compelling story for investors and for regulators. It's important to understand the mechanism of action of the drug. Even if the drug appears to work, we need to be able to show that we know why it works. And we need to demonstrate target engagement and ideally efficacy in a relevant animal model or models. Sometimes we have to consider developing an animal model because absence of one is not an acceptable excuse. We often need to develop biomarker programs that can be used in the preclinical and in the early phase clinical studies as surrogates or predictors of efficacy in the clinic. We need to demonstrate clearly the advantages of a product in vitro and if possible in vivo. And we need to demonstrate knowledge of the patient pathways and explain to investors and to regulators how the product will be used in the real world. Bearing in mind what I've said about the attrition rate during drug development and the need to attract funding, it's critical to plan with the end in mind, i.e. the end of the development program and commercialization. And the vehicle for doing this is the Target Product Profile, or TPP. The TPP is a document which needs to include all of the requirements required to define a successful product, i.e. its mechanism of action, the IP protection position, the patient population and market size, the intended product labeling, i.e. clinical safety and efficacy, what the likely warnings and contraindications might be, the regulatory plan for approvals and the launch strategy in the relevant markets, the commercial costs of goods, the competitor landscape, and the pricing and reimbursement strategy. We advise clients to start to build the TPP as early as possible, and certainly in parallel with candidate selection, because meeting a minimum TPP may directly inform the selection process. And in product development, it's important to fail fast before lots of time and money are potentially wasted. The TPP needs to be a living document, which should be reviewed and potentially revised on an ongoing basis as additional data is generated and in response to changes in the external environment. And actually, a TPP generated at any point before phase 2 is better than not having one at all. However, if at any point during development it's clear that the minimum TPP cannot be met, the program should be terminated. This is particularly difficult for single product companies to accept, which may be a reason that termination is put off. However, even with a single product, there are a number of strategies that can be adopted, e.g. having multiple clinical indications and therefore more than one TPP. So now we're going to talk about integrated program management. The difference between integrated program management and project management, be it non-clinical, clinical, regulatory project management, is because it's integrated. This needs an experienced program manager, ideally from a background in pharma or biotech, who has managed development programs previously and is able to align the planning with the TPP and plan the program years into the future. There's also a requirement to do scenario planning and look at different eventualities and the critical paths for those options and the impact of the changes on them. Not doing program management in this integrated fashion risks inefficiencies and programs taking longer than they should. For example, due to a lack of drug substance or validated assays, regulatory approvals taking longer than required, or a lack of funding, or a lack of CMC or non-clinical data because things have been missed, or an inability to partner and exit at the desired stage because of a weak IP position. Therefore, experienced program managers have to have substantial knowledge of most aspects of the drug development process and work with their plans on a daily basis, refining and challenging them, and most importantly, talking to senior management, functional and operational group heads, and program leads. This role becomes critical if the senior management or the founders are not from R&D background or experienced drug developers. So to recap, integrated development planning needs to be cognizant of all of the aspects of the program. It needs to be aligned with all the functional groups and the disciplines working on the program. It needs to be fully aligned with the target product profile. The timeline for the program is a living document that will be updated as the program progresses. When building timelines, integrated development planners often use software such as Microsoft Project to build Gantt charts or Smartsheet. And these tend to lead to a document of truth with hundreds of lines from which sections can be extracted and summarized into slide decks for presentation to senior management, boards, and investors. However, not everybody within the team uses the same software for creating these Gantt charts, and people often work in Excel, for example. But ultimately, this can only create a list of tasks and can't show the dependencies between the tasks or the impact of issues of one activity being delayed on subsequent or parallel tasks, for example. Also, it's difficult to interrogate the critical path, and one cannot easily link resources and costs to the activities. And for me, there's a particular problem with the schematic timelines people will often show in Excel and PowerPoint, where studies and activities are designated as blocks and arrows of time. This is crude, and it often leads to incorrect periods of time being allocated for study setup, closeout, reporting, or the hiatus between studies and the time needed for regulatory interactions. So overall, the duration of the project can be underestimated. So let's take a look now at some of the examples of the types of plans that people produce. This is an example small molecule non-clinical plan produced by Covance. And it serves overall to indicate that it is possible to complete an IND CTNA enabling package for a small molecule in 6 or 7 months. However, it can take longer than some people think to incorporate the data from stability studies or final non-clinical reports into documents such as the IND or the IMPD, to check that the text is aligned between the reports and summary documents such as IBs, complete internal reviews, and get all the approvals and signatures required ready for publishing and submission. Sponsors quite often underestimate how long this will take. And in small organizations, there are always competing priorities. And typically, if people think something will take a month, it may often take closer to 3. Also, don't underestimate the length of time it will take for review and approval. Not all agencies comply with their own stated or mandated review timelines, and there are often questions and requests for information, so several cycles of review are required. In the UK, the new Combined Competent Authority Ethics Committee approval process for clinical trials is helping, but this doesn't exist in all countries, and it means it's difficult to schedule site initiation visits. A few tips on other areas of the plan. Start your analytical method development and validation work as soon as possible, because this often takes longer than one might expect, especially if the CDMO or CRO you're using doesn't have experience with your particular class of molecule or a new technology. And utilize the ability to do quick, non-expensive, non-GLP, non-clinical work early on, such as Ames testing to get an early read on genotoxic potential, Hurg assays to give a read on potential cardiac issues, and importantly, what we call feed and bleed or PK studies in rodents. These can also give you an early read on the target organ or tissue toxicology, the feasible dose range, and also the PK profile of your compound, and can save time and money later if issues arise. What I would say is if these types of studies are being done within academic laboratories, it's important to get them written up properly because they will be supportive later on. This is a CMC plan for a gene therapy product and enables me to highlight some of the areas that require forethought and planning to avoid delays. These include work on the master cell bank production and working cell bank production to ensure that yields and titers can be reached, and to establish whether batch manufacture or continuous culture is appropriate. One also needs to establish and document the origins or the provenance and the intellectual property ownership and licenses required, covering all of the starting materials and key reagents, e.g. plasmid, payload sequences and other sequences, vectors, etc., which can save time when compiling IMPDs and potentially avoid issues with licenses having to be established in a hurry later. As much as possible, do GLP tox work using the GMP engineering batches, which should be representative of the clinical batches. People often think they have to actually do the tox with the clinical batches. This isn't the case. Engineering batches are usually used. So this is an example of an integrated development plan extracted from a Microsoft project Gantt chart. Now, one can have the CMC, non-clinical, and clinical and regulatory plans either fully expanded to reveal all the details and the subtasks, which can run into hundreds of lines in a development program, or rolled up to produce a high-level plan. But what Microsoft Project does do is to allow for the task dependencies to be properly reflected, which isn't the case when you show a development plan in Excel or PowerPoint. Remember that the task comments that you can add in project are not visible and can't be accessed in other formats. So if you want people to see these comments, which are quite often important, you need to circulate these prior to the meeting. And it's not always necessary to show the task bars. These are a useful visual representation, especially to show the overall duration of the project. But they're not necessary to facilitate a team discussion for which the task activities and the start and finish dates can be acceptable. So examples then of where more time than people think is often required will include moving from draft report to final report for non-clinical studies. And this is often dependent on the number of review and revision cycles required. And often sponsors have external consultants who also need to review. The same can be said in terms of finalizing protocols. Moving from a draft protocol to a final protocol, whether it's a non-clinical or clinical study, can take longer than people think. Also, in non-clinical studies, ordering animals, breeding of animals, and bringing them into the CRO and allowing them to acclimatize can add many weeks to the total duration of the study. And then if we move into the CMC arena, review of manufacturing records and batch records often takes longer than people think. Remember also that QP release is not just a perfunctory exercise. The QP who's releasing the batch for clinical trial use may have questions or issue requests for information, and they may even want to audit the manufacturing facility or facilities, which can add many weeks before the material is released for the trial. And then my final tip here is don't chase stability data. T0 or the analysis of the initial time point should really be within days of the date of manufacture. However, it can take longer to analyze subsequent pull points than people think, and it's not unusual for CDMOs to spend up to 4 weeks analyzing each pull point. And then it's usual that in a CTA application, at least a month's stability data is required in the IMPD, often 3 months. So get the stability studies started as soon as possible in advance of your submission. So integrated plans can also be used as a communication tool. I mentioned earlier that people often use Excel or PowerPoint to show timelines, but the danger here is that the overall timeline may be substantially underestimated. Also, it's not unusual that senior management have created a timeline based on what they want to happen to meet corporate goals versus reality. And the advantage of building your Gantt chart first is that you can discuss with the operational groups how long tasks will actually take and actually build the timelines with the correct amount of time plus a modest contingency built in. Then it's not guesswork, because otherwise one sets the company up potentially for failure or at least conflict within the organization when it becomes apparent that more time is required to complete a particular stage. One of the reasons I think this sometimes happens is because companies are keen to show investors a palatable timeline for a program through to the start of a clinical study or a readout or a particular endpoint. However, investors are now very used to the R&D process, and presentation of overly optimistic timelines simply damages credibility. So now we'll go on to talk a bit about risk management, which is the continuous process of identifying, analysing and responding to risk factors throughout the life of a project. There are 6 steps here I want to talk about. One is about identifying the risks. And risks usually have a negative impact on the prospect of achieving success. The risks need to be acknowledged and categorized and prioritized. Step 2 is about assessing the risks. Usually they're categorized by looking at the impact of the risk happens, if it's low, medium and high, and what's the probability that it will happen, even if it's a low risk. Is there a low, medium or high probability that it will actually occur? One then needs to interrogate the likely delay in terms of time or the additional cost or the additional resources which will be required if these risks come to pass, and the impact it will have on the quality of the product. Will the TPP actually still be able to be met? Important to develop a risk register that's discussed at regular team meetings so that plans can be put in place to try and avoid risks and eliminate a specific threat, maybe by removing the likely cause of those risks. Risk mitigations. So working out in advance what the company will do if certain risks come to pass. Risk mitigation or risk reduction is actually the same thing. And then risk acceptance. It may be that one is able to accept the consequences of the risk. And if that's the case, it's often accomplished by developing a contingency plan should the risk event occur. Owners should be assigned to each risk who take responsibility for working out how to try and avoid those risks, mitigate them if they occur, and build in the contingency planning required to deal with them if they occur. Step 5 is about communicating risks. Leadership needs to be aware about the critical risks within a program, and they need to be notified as soon as possible if it looks like something will go wrong or a risk will come to pass. And these needs to be tracked on a day-to-day basis. And this is one of the key jobs of the integrated program manager. So I now want to talk a little bit about the non-clinical program. In discovery, the focus needs to be on the preclinical science. We need to select the most appropriate clinical indication or indications. To do this, we need to consult widely with key opinion leaders like pharmacologists, clinicians, and the commercial people in the organization. We need to establish a proof of mechanism in vitro and, if possible, in vivo. This is a common roadblock for partnering. Even when it's been shown that the product has efficacy, we need to understand and communicate why and how it delivers this efficacy, because this also relates to off-target toxicology. And investors and partners put a lot of store in understanding the proof of mechanism. We need to understand what biomarkers, which are surrogates of clinical efficacy and safety monitoring, can be used in clinical trials. And we may need to develop appropriate animal models of human disease. There are many examples where this is very poor. For example, in neurodegenerative diseases such as ALS, Parkinson's, Alzheimer's disease, the available animal models arguably don't recapitulate the clinical etiology or progression of the disease. So it's possible that you will have to develop your own animal models. And ideally, during this phase of the program, one would build up an understanding of the ADME and PK profile of the compound to help select the dose ranges for GLP tox, and then in turn to select the most appropriate starting doses in man. It's more common now for phase 1 studies, first in human studies, for the starting dose to be based on MABEL, or the minimum biological effect level, which is a more conservative approach, as opposed to the NOELs and NOALs, which can overestimate safe doses. And obviously, species selection. This is a key thing, and it's very important to select the most appropriate species. And there is some latitude within the regulatory guidance. Obviously, the standard procedure is 2 species, a small animal or rodent species and a larger animal species, which tend to be rat and dog, but alternative approaches can be justified. It's important during this phase of the program to build a robust testing cascade, which will pay dividends later. A testing cascade will help to select the front-running molecules during candidate selection. And many of these tests can be conducted in parallel or sequentially if there are financial constraints based on a testing hierarchy, with the most important being conducted earliest. One can optimize these to be cost-effective, running the least expensive tests in-house first, and then making decisions on any that need to be progressed externally. And in vivo studies in whole animals should only be pursued for molecules which have successfully passed or met the criteria from in vitro testing, which helps to minimize the use of animals in compliance with the 3Rs principle. When designing an efficacy and safety testing cascade, one typically considers those tests that can be conducted in-house, such as enzyme or cell potency assays, selectivity assays, ADME studies, and initial non-GLP tox studies, to see whether the PK profile can be achieved and to look for overt signals of toxicity or genotoxicity, e.g. in Ames tests, and also to generate some proof of mechanism data in whole animals or appropriate disease models. It's important when profiling compounds to have the required assays and biomarkers for safety and efficacy in place. And ideally, one should use commercially available reagents or reagents and methodologies that can be easily transferred, for instance, from an academic environment into commercial CRO labs, and be adapted to run under GLP or GCP conditions. Going forward, smarter in silico testing and the decrease in the use of whole animals for non-pivotal safety work will become standard, and this needs to be recognized and adopted. We'll now take a look at the regulatory guidance covering the non-clinical program. And there is much regulatory guidance, which differs according to the type of products being developed, and is well established now for small molecules and large molecules. And increasingly, there is more guidance being issued by EMA and FDA for cell and gene therapy products. However, with novel technologies such as extracellular vesicle technologies and nanomedicines, there is still relatively little guidance compared to the other types of product, and this will continue to evolve. Typically, in ATMPs, the ADME program is replaced by biodistribution and shedding studies. And typically, integration is looked at instead of the traditional genotoxicity studies. Now, some non-clinical data are clearly mandatory prior to a first-in-human clinical study. But some other studies are only required before larger clinical studies, i.e. prior to phase 3, such as chronic toxicology and carcinogenicity studies, which are very much related to the duration of dosing in later phase clinical studies, and also whether or not any genotoxic findings have been determined earlier during the non-clinical program. And then again, some studies are required before women of childbearing potential can be enrolled into clinical studies, such as reprotoxicology studies or oral contraceptive interaction studies, or before children can be enrolled, such as juvenile tox studies. As I stated earlier in the presentation, it is possible to propose alternatives to the studies or the package of studies outlined in the guidance, if there's a strong scientific rationale which can be justified robustly. And we urge our clients to discuss their proposed non-clinical programs with at least one of the major regulatory agencies before embarking on an IND or CTA enabling program. Now, the non-clinical program is designed to support the first-in-human strategy. The first-in-human studies, typically with small molecules, are in healthy volunteers, SAD, MAD, food effect type studies. But increasingly, we're seeing first-in-human studies being conducted in the patient populations, phase 1, phase 2A patient studies, e.g. in oncology, in surgical indications, and with ATMPs in rare diseases. Now, this is because cell and gene therapies are not usually able to be administered to healthy volunteers. Also, in the patient populations, there may only be a single dose administered therapeutically, and often the route of administration cannot be approached in healthy volunteers, e.g. subretinal injections or intratumoral injections. Now, clearly, the non-clinical program needs to be able to determine and predict any undesirable toxic and target organ effects in the animals at relatively large doses, but also at doses which are relevant to the clinical situation. And we need to understand the exposures which are relevant to these events and also whether they are reversible. We also need to define the ADME profile, or the biodistribution and shedding profile for gene therapies with viral vectors. And clearly, the whole point of the non-clinical program is to inform and support the first-in-human early-stage clinical trial designs. Toxic doses and safe doses in animals can be converted into human equivalent doses, and these levels can help to define the starting dose and also the maximum dose that we're likely to go to in the early clinical studies. We also need to identify the safety parameters and the biomarkers which will be measured during the clinical studies. So it's critical when planning a non-clinical program to have at least an outline study design for the first-in-human studies. Critical elements being a protocol synopsis at least, route of administration, doses, intended dose duration, pharmacodynamic or efficacy endpoints, and bioanalytical or biomarker readouts. These need to be understood prior to conducting a pivotal non-clinical program so that the appropriate studies can be done to support the intended clinical program. And if one wishes to discuss the non-clinical program with the regulatory agencies, as I've always advocated, then one needs to provide the agency with a protocol synopsis at the same time. Otherwise, they can't actually comment on whether it's applicable or not. And one thing to remember is that switching clinical indications for various reasons later may require additional non-clinical work to be done. Now we're going to turn our attention to the CMC program. Now, investing sufficiently and early enough in the CMC program, or rather not doing so, is a common issue we see in development. CMC, I think, is sometimes viewed as perfunctory, but in reality, it's often the source of issues in efforts to open an IND, and even much later, post-submission of an MAA, Problems often occur during pre-approval inspections of manufacturing facilities. It's critical for integrated development planning that one understands what the CMC program is going to deliver and when. For example, the amount of drug substance required for repeat dose tox studies in large animal species can be significant, and I mean kilo scale. So this means that we need to work on the scale up, often from the milligrams, the tens of milligrams in the lab, to transfer the process to a CDMO or a CMO who can scale up the process and optimize it to work at the kilo scale. This often takes a lot of time. We need to increase the purity of the drug substance over time, and we need all the potential related substances, these are the impurities and degradants, to be present in the pivotal tox studies. If the material used for pivotal tox is very pure, there's a risk that other impurities will be found later on in clinical batches. So it's best to use a batch of drug substance or batches of drug substance which do have some impurities and can be improved on for the clinical batch. Biocompatibility studies are also sometimes not thought about, and this is really to look at the interaction of formulated drug product with plastics and other materials, especially those used in the clinic, tubing, syringes, etc. And that may require the generation of extractable and leachables data. People often forget this until the last minute, which causes delays. So we advise clients to plan early and plan the assays that they will need in the CMC program early, because they can be time consuming and require rework. Try and plan non-GMP and GM batches in campaigns to save money. And plan formulation and stability studies early to save time. And we've talked already about the importance of engaging with the regulatory agents. Discuss the IND or CTA enabling non-clinical program, which accounts for the majority of the questions raised in scientific advice meetings with EMA. Most people know that this is typically done in a format where we provide a briefing book and ask questions to the agencies. What is less commonly understood is that the company position has to be included, i.e. you need to answer your own questions and support your own proposals to do what you want to do, and then ask whether the agency agrees, or be prepared to be advised that they do not agree and hopefully recommend a different approach. Sometimes they expand on this to a greater or lesser degree. So example general questions are, does the agency agree that the product specifications for product X are suitable for phase 1 development? Or does the agency agree the planned and completed non-clinical studies are acceptable to support the initiation of clinical trials with X? And obviously in your proposals, you will have answered your questions and provided a justification for the program that you want to run. So a little word about types of regulatory meeting. The major agencies, and I'm covering here just the MHRA, EMA and FDA, have various meetings throughout different stages of the development program. And early on, people will either usually take pre-IND or scientific advice meetings. However, there are earlier stage meetings available with MHRA Innovation Office meetings, Innovation Task Force meetings with EMA, and with FDA, the Interact meetings. Now, the choice of which agency meetings to request and which type of meetings depends on where the intended focus of the development is geographically, and also the nature of the product type or class and the technology it's based upon, and how well this might be understood widely and within the agencies, and whether you're looking for formal advice or confirmation of your plans, or just an initial discussion. With cutting-edge technology, it's a good idea to have an innovation type meeting to familiarize the agency in a slightly less formal manner before much data has been generated, rather than the formal scientific advice or pre-IND meeting, which needs a lot of data to have been generated before the meetings go ahead. In any situation, high-quality briefing books are critical. If we look a bit later into the program, there are a variety of regulatory interactions, some very, very important ones, and some others less so. One thing I would advise is be prepared for conflicting or overlapping advice. It's still the case that the agencies are not completely aligned on some topics, and so it's always best to actually take advice from perhaps the US and Europe if one is considering a parallel track development in those major markets. Now I'm going to talk a little bit about the designations that one can apply for from the regulatory agencies to give extra protection or commercial viability to a product. The first of these is the orphan drug designation. As the name suggests, this covers drugs which are intended to treat patients with a rare or orphan disease, and over 6,000 of these diseases are known, affecting up to 1 in 17 people, for example, in the EU. And the main criteria are based on the prevalence of the disease. There has to be a scientific rationale for use in that orphan condition and various levels of data. Now, the rules governing eligibility in terms of what type of non-clinical and clinical data are required to achieve these designations do vary in the different jurisdictions. Orphan drug designation doesn't actually block competitors from developing a different product in the same indication, or even a very similar product in a different indication. But it does block a similar product from being approved in the same indication. But there are caveats around this. The orphan drug designation can be useful commercially to apply for, and if it's designated, it does offer extra market exclusivity, for example, 10 years in the EU as opposed to 7 years in the US. Now, there are a range of other schemes which are intended to assist sponsors with their development programs and give them access to agency input to increase the likelihood of the program progressing successfully. Now, these don't accelerate development per se, but they can be associated with accelerated review timelines. I've highlighted here PRIME in the EU, Breakthrough Designation in the US, and the ILAP in the UK. And these designations are perceived as valuable by investors, and in operational terms, they're also very valuable. Again, like the orphan drug designations, there are different eligibility rules in different jurisdictions based around new therapy modalities that have the potential to change the status quo more than just an incremental impact over and above standard of care, or to address areas of high unmet medical need. These are the common factors. And it's true to say that in Europe, the vast majority of prime applications granted so far have been for oncology products. Now, some final thoughts about regulatory considerations as part of an integrated development plan. Work backwards from the clinical plan when it comes to your non-clinical program. Ask the questions: what is the patient population that we're going to be treating in the clinic? Who will be exposed and treated? By what route? How often? And how long for? Consider whether it's necessary to follow the regulatory guidance to the letter, or whether there's a scientific rationale as to why something's not feasible or applicable. And this may be related to the type of medicinal products that you're developing, which may mean there is a strong rationale to do something that's not in the guidance or to do something in a different way. Try and use your regulatory intelligence to predict and address potential problems before they become an issue. Use vehicles such as public assessment reports, and in the US, the summary of basis of approvals, and AdCom meeting minutes to try and work out the hurdles which your product is likely to encounter, and discuss with regulators through the scientific advice route and other earlier stage agency meetings, because they're there to help. And above all, plan your regulatory strategy in your agency interactions and build them into the overall integrated development plan. And make sure that the data will be available when they're needed for those agency interactions. Okay, so now some top tips from the frontline. Don't underestimate the value of non-GLP, non-clinical studies and PK studies in rodents to help understand the PK/PD profile of your compound administered by different routes, and to gain some early indication of target organ toxicity and adverse signals. Consider conducting AIM studies and HERG studies early on, and also pre-formulation studies even before you've manufactured GMP material, and ensure they're written up and reported correctly, especially if they're undertaken in an academic environment. Please invest upfront in assay or method development and validation. One often encounters technical difficulties with these studies, and they always take longer than planned. Initiate a rolling program of API manufacture and scale-up to supply the CMC requirements of your program. Remember, repeat dose tox studies in dogs and non-human primates in particular consume a lot of material. Ensure that the synthetic route and the manufacturing process has been optimized to the extent you can at the scale you're working in, and that you understand the impurity profile and can quantify the impurities before embarking on pivotal tox studies. For ATMPs, use GMP engineering batches to supply the tox studies. As long as they're representative of the clinical batches, they don't need to be GMP. However, they should never be more pure than the clinical trial material. Don't chase stability data, and build in sufficient time to generate that data prior to regulatory submission dates. One needs a minimum of 1 month and sometimes 3 months on different batches to support a clinical trial application, depending on the phase. To avoid difficult discussions with regulatory reviewers, minimize the lag phase between the date of manufacture of batches and putting them up on stability, or the T0 or initial time point, and ensure that any out-of-scope results are fully investigated and written up prior to dossier submission, because questions will arise. For temperature labile IMPs, think about the need for benchtop stability studies to cover the thawing of frozen material and manipulation of IMP at ambient temperature between removal from controlled storage and administration to subjects in a clinical trial. Remember, tricky CMC questions will always arise from regulatory reviewers during CTA and IND reviews, and represent the most common reasons for hold-up and grounds for non-approval. So pay attention to the CMC aspects of your program. They're at least as important, if not more important, as the non-clinical and the clinical aspects. It's been a pleasure talking to you today, and if you'd like to get hold of me or any of my colleagues at Boyd's, please contact us. Thank you for listening.